All right, guys, so here we are in our new section, sections 5.1 and 5.2, the cell cycle and mitosis. Our key concepts, we have two of them. Cells have distinct phases of growth, reproduction, and normal functions, and cells divide during mitosis and cytokinesis. We have a lot of vocabulary and a lot of crazy vocabulary, but don't worry, we'll get it. So we have the cell cycle, interphase, gap 1, which we abbreviate G1, synthesis with an S, gap 2, again abbreviate G2, G0, mitosis, prophase, metaphase, anaphase, telophase, cytokinesis, apoptosis, cancer, benign, malignant, and carcinogens. Our essential questions are: what are the main events of the cell cycle? What events occur during each of the 4 phases of mitosis? How do daughter cells split after mitosis? How is the cell cycle regulated? And how do cancer cells differ from other cells? So we're going to start writing our notes here. So the cell cycle. So basically before a cell divides to make more cells, it goes through a pattern of growth and copying DNA, and then finally cell division. We call this process the cell cycle. So basically, like I said, it goes through this process every time it's going to make more cells. So the cell basically grows up to a certain point. It copies the DNA, remember DNA replication. Once that's done, it gets ready, it starts to divide, where the cell starts to go in, split on itself, eventually forming 2 new cells. Those 2 new cells do the same thing. They grow, copy DNA, get ready for dividing, divide, do, 2 new cells, okay? This happens in all of our somatic cells. Somatic cells, remember from our presentation yesterday, somatic cells are your body cells. So your blood cells, your skin cells, your bone cells, all those cells are your body cells. The other cells in your body are your reproductive cells, and those are the sperm and the egg. So the mitosis or the cell cycle happens in all of your body cells. We have 4 more main stages. We have gap 1, again, abbreviate G1, synthesis, which we abbreviate with an S, gap 2, which is G2, and then mitosis. So here's just a picture showing you the cell cycle. Now you'll notice that most of the time is spent in G1, and we've got S and G2. That takes up the longest amount of time. The other part, the M, there's not a lot of the piece of the pie that is spent in that time. So if we look at this a little bit closer, you can really see that most of our time is spent in that growing, the copying of the DNA, and being ready to divide. The cell division part only takes up a small amount of the cell cycle. All right, so the first 3 stages of the cell cycle, those longest ones, we group into a category called interphase. In this part, the cell is just basically getting ready for dividing. So the cell grows and it copies its DNA and it prepares for division. 95% of the time of the cell cycle is spent in interphase. And that's a common question for you to be asked on like your EOC at the end of the year or possibly even your midterm. They'll ask you, which is the longest part of the cell cycle? It's interphase. The cell's getting ready to divide. It takes a long time for it to get ready. We have 3 stages that make up interphase. We have gap 1, we have synthesis, and gap 2. Gap 1, if you can remember, the G just stands for growing because the cell is just growing. Synthesis. Synthesis means to make. So in this part, we have a replication of DNA. It's making more DNA. It's copying that DNA. And remember, when it copies the DNA, it's copying that whole chromosome. So when the chromosomes get stuck together, they're identical copies. Then we have G2, G again standing for grow. So the cell's growing and organizing itself for division at this point. Next, we have mitosis and cytokinesis. Now, mitosis it's basically like the cell splitting the nucleus into 2. Okay, it's not when the cell actually moves away from each other. That part's called cytokinesis. But mitosis is the division of the nucleus. Only 5% of the cell cycle is spent in mitosis and cytokinesis. We've got 4 major phases of mitosis. And if you can remember, PMAT is going to help you keep track of the order. So PMAT, we've got prophase, metaphase, anaphase, and telophase that make up mitosis. And then finally, after mitosis, we have this process called cytokinesis. Cyto, cell, kinesis, moving away from. So it's the cell actually splitting into 2 distinct daughter cells. So cytokinesis is the division of the cytoplasm. The cell finally splits, dividing into two genetically identical diploid cells. Okay, diploid, 2N, two copies of each chromosome. Okay, we're going to talk a little bit closer about the different stages of the cell cycle now, or sorry, mitosis now. So the first stage of mitosis is called prophase. In prophase, our cell is doing some things here. So genetic material condenses, forming those actual chromosomes. We see those nice little X's sitting there in the middle of the cell. So our duplicated chromosomes become very visible. The outer membrane that surrounds the nucleus, called the nuclear membrane, starts to dissolve, so those chromosomes can actually come out of the nucleus. Outside the nucleus, we see these little spindles beginning to form. The spindles are like little ropes that are actually going to grab a hold of the chromosomes in a little bit. Then we've got metaphase. Metaphase, if you can remember meta for middle, because the chromosomes are going to line up down the middle of the cell. Okay, so you see this nice little line of them lining up down the middle. Those spindle fibers, which look like little ropes, connect to each of the chromosomes and they attach by, in the middle with that centromere. The centromere is what connects those two sister chromatids together. Remember, this side is identical to this side. So eventually what's going to happen is they're going to split apart, okay, moving along the spindle fibers. But in metaphase, if you remember meta from middle, because the chromosomes are lining up down the middle. Then we've got anaphase. Anaphase, those chromatids are actually moving away from each other. Okay, so ana for away. So chromosomes separate and move along the spindle fibers to the opposite ends of the cell. Again, anaphase, now those sister chromatids are going to move away from each other to opposite ends of the cell. Okay, then we have telophase. In telophase, now the sister chromatids have moved away. Remember, this is exactly the same as this. They're exact copies, moving away from each other. Two nuclei form. Nuclei is just plural for nucleus. So we have one nucleus on this side and one nucleus on this side. So they start to form. So we start to see that nuclear envelope, what surrounds the nucleus, starting to form, forming around those chromosomes. The chromosomes begin to uncoil, turning back into their chromatin form, which looks like it's just a big pile of string. The spindles start to break apart and shrink down. And finally, mitosis is complete. We have two nuclei. So if you can remember telophase, in telophase we have two nuclei. It's plural for nucleus. So a nucleus on this side and a nucleus on this side. Finally, we have cytokinesis. In cytokinesis, this is where the cell actually divides into two genetically identical diploid cells. Okay, cytokinesis differs in animal and plant cells. So we look at this picture up above. I've got the picture of we've got our two distinct daughter cells. They're genetically identical in every single way. Okay, so mitosis makes absolutely identical cells. Because in your body, if you're making skin cells, you want those skin cells to be exactly identical to the first ones that they came from. Okay, so if you start with a certain number of chromosomes, say we start with 10 chromosomes in our first cell, we are going to end up with 10 chromosomes in each of our daughter cells. If we start with 5 chromosomes in our first parent cell, we're going to end up with 5 chromosomes in each of our daughter cells. Okay, they're making genetically identical cells. Now, like I was saying, cytokinesis differs in animals and plant cells. In animals, they're kind of like the cells are soft and squishy, right? So when the cell is getting ready for dividing, it's going through mitosis. Towards the end, when telophase happens, it kind of pinches in on itself, pinches until finally it closes up. Cytokinesis occurs where the cells are moving away from each other. Plant cells have that cell wall. They're very rigid. They cannot pinch from the tops and the sides, okay? So what they do is they actually form a little wall in between, okay? It's called the cell plate. This little wall helps then to push those two cells apart, okay? So it's a little bit different. So please, in mitosis, remember this. After cells divide through the process of mitosis, all cells have the same number of chromosomes as the parent cell. It produces two identical diploid daughter cells, okay? And again, if our first cell starts with 100 chromosomes, our two new daughter cells are identical. They are going to have 100 chromosomes. Now, cells do divide at different rates, like your skin cells divide more quickly than a lot of other cells in your body. Some cells hardly ever divide, and they sit in a stage called G0. Basically mean they're not going to divide, okay? G0, nothing's happening. Things like your brain cells, okay? Nerve cells take a very, very long time to regenerate and to grow back and divide, but skin cells only take about 2 weeks, okay? Now, cells actually don't just keep grow, okay? They have to go through this process of division. And cell growth is coordinated with division, and the volume of a cell actually increases. So like the inside, okay, the volume increases faster than the surface area. Cells that have to be large have very unique shapes, okay? So you can see this. So it's easier for them to move things in and out of their cell. We don't want them to be too big, because if they get too big, it's very hard for cells to move things in and out of them. Now, there are sometimes problems in the cell cycle. And actually, during each part of the cell cycle, there are these little checkpoints. It's kind of like the police. They go around and make sure everything's happening the way it's supposed to. And checkpoints in the cell cycle will prevent cell division from happening if maybe the cell doesn't have enough nutrients for it to happen. Maybe the cell's not big enough, it didn't grow fast enough, or if the DNA hasn't been replicated or is damaged. So there's these checkpoints, like the little police that come in and say, okay, are you ready to divide? Are you sure? Do you have this? Do you have that? Uh-oh, nope, you don't have that. Well, sometimes things can be corrected. So the cell will actually go back to one of the stages before it and maybe copy the DNA or grow a little bit more and then start dividing. Sometimes there's such big problems that nothing can be done. So the cell has to go into this process of actually dying off, or it's called programmed cell death, which is called apoptosis, where the cell actually divides. It's told, hey, you cannot be here. You are so messed up. So it goes through this process of apoptosis, which is a programmed cell death. And this is just a little thing showing where each of these checkpoints occur. One happens right after G1, one happens right before mitosis, and then one happens again right before cytokinesis. And again, it's just to make sure that everything is where it needs to be, that the cell is doing what it's supposed to in order to divide. Because once that cell divides, it's making 2 new cells. And those 2 new cells are going to go on to divide to make 2 more cells and so on. So we don't want any problems. Now, sometimes mutations happen. Remember, mutations were changes in the DNA, causing some cells to sneak through these checkpoints, okay? When that happens, it can cause an out-of-control cell cycle, where the cell cycle is sped up, causing cells to grow out of control. They're not being checked anymore, okay? They've been very sneaky. They've passed these checkpoints. They no longer have to go through them. The result of uncontrolled cell division is called cancer, and you've heard of cancer. Cancer cells can be benign, which means the tumor is relatively harmless, okay? The cells are just growing out of control, making a little bump, okay? So it's benign. Sometimes it can be malignant. Malignant is when the cancer cells can break away and spread to other parts of the body. And the spread of cancer is called metastasis. Lastly, carcinogens are substances that are known to cause cancer, and you guys know about a lot of these. We talked about them when we talked about mutations. So there are different chemicals in the environment that can cause mutations, different substances, UV rays from the sunshine. There's a bunch of different things that can be considered carcinogens. And again, the out-of-control cell cycle, the result can be cancer, and cancer can be benign or malignant. Okay, so this is it for the cell cycle and mitosis. Please, guys, please remember mitosis produces genetically identical daughter cells. So 2 genetically identical daughter cells. Those daughter cells are diploid. They have copies from mom, copies from dad. Okay, if we go into the process of mitosis and we start that process with 100 chromosomes, those 2 new daughter cells are going to each have 100 chromosomes. You will be asked that on your midterm and your EOC. So remember, mitosis, genetically identical daughter cells. More specifically, 2 genetically identical diploid daughter cells. If you have any questions, please reach out. Let me know. Thank you.